The ability to sense heat is one of the body's most vital protective mechanisms. It keeps us from touching a hot stove, warns us of danger, and helps us avoid injury. But for decades, scientists have puzzled over a fundamental question. How does the body actually detect temperature at the molecular level? Recent research from Northwestern University has finally provided a detailed answer, offering a molecular snapshot of how our bodies know when it is too hot. Temperature is an ever-present environmental factor that shapes our daily experiences, from the warmth of sunlight to the sting of a burn. Yet, the process by which our bodies translate temperature into sensation has remained elusive. Unlike drugs, which can be tracked as they bind to proteins, heat has no physical form or binding site. This has made it notoriously difficult for scientists to study how our cells sense temperature changes. The mystery has persisted, leaving a gap in our understanding of pain, healing, and disease. At the heart of this discovery is a protein called TRPM3, which stands for transient receptor potential melanostatin 3. TRPM3 sits in the membranes of cells, acting like a tiny gatekeeper. When it detects heat, it opens up, allowing charged particles known as ions to flow into the cell. This flow of ions triggers nerve signals that travel to the brain, where they are interpreted as sensations of heat or pain. TRPM3 is not just a heat sensor. It also plays a role in pain, inflammation, and even epilepsy, making it a crucial player in the body's sensory system. Studying how TRPM3 works required innovative techniques. Since heat cannot be seen or directly tracked, the research team led by professors Huan Du and Wei Lu turned to cryo-electron microscopy, or cryo-EM. This advanced method involves flash freezing proteins and taking thousands of images to create detailed three-dimensional models at near-atomic resolution. Alongside cryo-EM, the team used electrophysiology, a technique that measures electrical currents passing through proteins in living cells. By using a chemical that mimics heat, they captured TRPM3 in its active state. Then with an epilepsy drug that binds to the protein, they observed its inactive state. Comparing these two states revealed which parts of TRPM3 shift during activation, providing a foundation for understanding how the sensor responds to heat. One of the most surprising findings was that TRPM3 senses heat from within the cell, not from the section embedded in the membrane as previously thought. The protein acts as a molecular switch made of 4 parts. When the inner regions of these parts are tightly connected, TRPM3 remains inactive. Heat, or a chemical activator, disrupts these connections, flipping the switch and activating the channel. Both heat and chemical activators push the same internal switch to activate the channel, said Huan Du. In contrast, the epilepsy drug jams the switch, preventing it from changing shape. This discovery uncovers a new way that cells sense temperature and helps explain how the nervous system distinguishes harmless warmth from dangerous heat. Because TRPM3 is found in both the brain and sensory neurons in the skin, understanding its function could have far-reaching implications. When TRPM3 becomes overactive, it can cause pain. By learning how this sensor detects heat and how to control its activity, we may discover new pain relief strategies that are safer and less likely to cause addiction, said Wei Lu. This research could pave the way for non-addictive treatments for chronic pain, inflammation, and neurological disorders like epilepsy. The molecular snapshots captured by the Northwestern team mark a major step forward in our understanding of heat sensation. As scientists continue to unravel the secrets of TRPM3, new therapies for pain and disease may be on the horizon, offering hope for millions who suffer from chronic conditions.